Welcome to Easy Bio. Today we are going to complete the plant nutrition. The points which needed, the limiting factor and how it changed according to the change in the environment, and the enrichment of the different factors in greenhouse system, leaf structure and mineral requirements for the plants. We'll begin with limiting factor. The definition is a factor which controls the rate of overall process, or the factor which restricts the effect of other factor affecting the rate of overall of process. What does it mean? We have an experiment, the plant, and we have light source, and they measured the distance between the light source and the plant. They make the whole factors available according to the natural environmental condition. So for this plant we have natural environmental condition plus the light source. They start to move the light source towards the plant, and they measured the rate of photosynthesis. Firstly, they found that the rate of photosynthesis increased by decreasing the distance between the light and the plant. Why? Because by decreasing the distance, we are increasing the light intensity, so the rate of photosynthesis will increase. But at certain point, they found that after this point, although we are increasing the rate of increasing the light intensity or decreasing the distance between the light and the plant, they found the rate of photosynthesis start to be constant. Why? Here I have increased the light intensity, but we have other factors. We have other factors which are naturally found in the environment, like carbon dioxide, which found in the environment with the percentage 0.04%. So I'm trying to increase the light intensity, but the rate of photosynthesis starts to be constant because we have limiting factor which is called carbon dioxide. So this carbon dioxide restricts the effect of the other factors, especially the light intensity. We are increasing the light intensity. We have chlorophyll in the plant. We have everything except the percentage of carbon dioxide, which is constant, and it restricts the effect of others. That's why carbon dioxide is limiting factor. So we have limiting factor which can be different according to the availability of the other factors. For example, if we have in winter, we have light, we have chlorophyll, we have carbon dioxide, but the temperature is less. So in this case, our temperature is the limiting factor, because the temperature less affects the rate of photosynthesis, so affect or restricts the effect of other factors. During night, we have chlorophyll, we have carbon dioxide, we have temperature, suitable temperature, but the light intensity is less. So in this case, light intensity will be the limiting factor for the other factors. Okay, so the availability of most of factors except one, this one restricts their effect on the rate of photosynthesis, which is called control, or which is called limiting factor, because it limits the overall process. So can we control this limiting factor? Actually, yes, we can control the limiting factor by the greenhouse system. We are making some enrichment for these factors, so the availability of factors will be there. So we don't have any limiting factor. So in greenhouse system, we have carbon dioxide enrichment. How can we supply the greenhouse system by carbon dioxide? By burning of fossil fuels. So carbon dioxide found in high percentage. Light, by using during the night, we don't have any problem because we have the sunlight, but during night So during day we don't have any problem because we have sunlight, but during night we have artificial lights. Temperature, using the heaters to adjustment the temperature according to the rate of photosynthesis. So these are the limiting factor. Then we need to know the structure of the leaf. This is the leaf structure, the internal structure of the leaf. We have the upper layer, which is cuticle, and it's waxy. It's waxy. So what is the function of cuticle? It reduces the water loss by evaporation, and you have to know that it just reduces, not prevents. So evaporation takes place, yes, but the rate of evaporation is lesser. The rate of evaporation is less. So we reduces the rate of evaporation to reduce the water loss. After that we have another layer, this one, which is called upper epidermis. Upper epidermis acts as like your skin. It's one cell thick. The thickness is only one cell thick. It's transparent. What do we mean by transparent? It means there is no chlorophyll at all, and this point is very important, you should know. There is no chloroplasts in upper epidermis. Okay, it allows, because it's transparent, so it allows the light to pass through it to reach the next layers for photosynthesis. So what is the function of it? The function of it acts as a barrier against the microorganisms like bacteria and viruses. So here we have the upper epidermis, followed by palisade mesophyll. As you see, the cells are longer or elongated or columnar. It means they are long and arranged in columns, as you see. And by the way, palisade mesophyll, as we said in the cell structure, is adapted or specialized by presence of a lot or many of chloroplasts. Many of chloroplasts here, as you see, many of chloroplasts in palisade mesophyll. What does it mean? It means the highest rate of photosynthesis. It means the highest rate of carbon dioxide absorption. It means the highest rate of production of oxygen. Okay, because he change or modify the question according to your understanding for this point. We have high rate of photosynthesis. It means high rate of absorption of carbon dioxide or production of oxygen, which is our waste product. After that we have spongy mesophyll, and as you see, the cells are arranged and between them we have some air spaces. So what is the function of this? Just to exchange the gases. We have one product released from palisade mesophyll, which is oxygen, and we have another gas which comes from outside the leaf, which is called carbon dioxide. The exchange of gases takes place in spongy mesophyll. Here I have vascular bundle, which is divided into two parts, xylem and phloem. This for water transport and this for food transport. The food which is glucose, which is produced by photosynthesis, is transported or translocate by the phloem. Finally, we have the last one, which is lower epidermis. Lower epidermis. In lower epidermis, we have a special structure which is called stomata. The stomata opens and closes according to the light intensity. According to the light intensity. That's why light intensity is very important for the photosynthesis. So when stomata opens, it allows the entrance of carbon dioxide and releasing of oxygen. Okay, so opening or closing stomata controls the rate of photosynthesis according to the rate or the percentage of carbon dioxide. This is the shape of, or this is the structure of stomata. Stomata, it has inner thick wall, it has outer thin wall and some guard cells, and we have here chloroplasts. We have chloroplasts. So do we have chloroplasts in lower epidermis? Yes, in the whole layers of the leaf we have chloroplasts except the upper epidermis. So here, and this is closed, as you see here it's closed, but here it's opened. Closed, controlled, as I told you, controlled by light intensity, but sometimes controlled by the water itself. If there is shortage of water, the cells or the guard cell or the guard cell becomes flaccid. So this guard cell leads to closing of the stomata. It affects the absorption or the rate of absorption of carbon dioxide, so it affects the whole or the overall process. That's why the water is important. The next point, mineral salts requirements in the soil for the plant itself. Actually, we have different mineral salts, but according to our syllabus, we need only 2 ions which are needed and their function and their deficiency. The magnesium ion required for synthesis of chlorophyll. Can imagine without magnesium there is no chlorophyll at all, so it will affect the plant. How? We'll explain later. Nitrates required for building up of amino acids. What are the amino acids? They are the simple units of protein, and protein used for the growth, used for the enzymes, building up of enzymes. So they are very important for keeping the plant healthy. So how their deficiency, how they affect the plant. For the chlorophyll or for the magnesium ions, it affects it by a disease which is called chlorosis. As you see, the plant completely healthy except the color of the leaves. So the stem is normal, the roots are normal, but we have the leaves which are turned into pale green or yellow, but we have normal roots. In case of the plant nitrates deficiency, the plant becomes stunted with weak stem, yellow dying leaves. Look at the plant, compare between it. Here we have magnesium deficiency and we have nitrates deficiency. Look, the roots slightly affected, but there is effect on the root itself, and the stem become very thin, and as you see, lower leaves dead and the upper leaves are pale. That's why the nitrate is very important.